Hello and welcome to this lesson for Advanced Higher Biology, which today is going to be looking at the control of the cell cycle. So prior to now, we've looked at the cell cycle itself and the stages within it and what's happening at each stage. Today we're going to be looking at how those stages are controlled before we move on in the next lesson to look at apoptosis or programmed cell death. So what you need to be able to do is understand why the cell cycle must be controlled and the processes which do this. So you're expected to be able to explain what is meant by a checkpoint in the cell cycle, explain the processes which occur at the G1 and G2 checkpoints, explain what is being checked for at the metaphase checkpoint, and state the problems caused by uncontrolled cell division, and describe the mutation of a proto-oncogene. So to begin with, we'll consider why we need to control our cell cycle. The timing and rate of cell division is crucial to normal growth, development and maintenance of tissues. The frequency of division itself is going to vary with cell type. So for example, human skin cells are dividing throughout life. Liver cells down here are only going to divide if the need arises, such as some kind of injury occurs. And in a mature adult, nerve cells do not divide at all. So the cell cycle needs to be controlled to ensure that all of the events proceed in the correct order and that each event is essentially finished before the next one starts. For example, we couldn't enter G2 and the M phase before we've managed to replicate our DNA in S phase. We also need to make sure that there are sufficient nutrients and growth factors in the cell before it can commit itself to the next stage in the cell cycle. So there's a few checkpoints at various stages, and this is where we have stop and start signals which are regulating the cycle. A checkpoint is essentially a mechanism within the cell that's assessing the cell. So it's looking at its condition and it's going to halt the progression into the next phase until all of the appropriate requirements are met. And there's three major checkpoints in the cell cycle. We've got the G1 checkpoint, the G2 checkpoint, and the M checkpoint. Our first checkpoint is our G1, and at this point we are looking for an appropriate cell size, so sufficient cell growth must have occurred, and we must also have appropriate amounts of nutrients and growth factors before we can move into S phase. Now there's a few proteins involved in this one. We have ones which are referred to as cyclin proteins. They accumulate during cell growth and are involved in regulating the cell cycle. So a cyclin will bind with another protein which is referred to as a cyclin-dependent kinase, or CDK. And if you remember, a kinase enzyme is any type that's going to phosphorylate something. So when we have cyclin present, it's going to bind to the cyclin-dependent kinases. An active cyclin CDK complex will phosphorylate, therefore, different proteins that regulate progression through the cell cycle. Once we have a sufficient threshold of phosphorylation, the cell cycle can move on to the next stage. If we don't have a sufficient threshold, then the cell is going to be held at that checkpoint until we can reach the correct amount of phosphorylation. So under normal circumstances, or the beginning of the cell cycle, we don't have any cyclin produced. That protein has not yet been synthesized during the G1 growth phase. As the G1 growth phase continues and we produce enough cyclin, that cyclin comes along and activates our CDK protein. The CDK protein is then going to be responsible for phosphorylating all these different target proteins, and they're going to help continue to phosphorylate, and our specific targets mean that once we reach enough phosphorylation, we're then able to move on to S phase. We also have another protein involved, which is the retinoblastoma protein, or RB protein. Now, this normally acts as a tumour suppressor, and what it does is it inhibits transcription of genes that code for proteins needed for DNA replication. So if you remember, we're moving into S phase. We need DNA replication, we need to replicate all of those chromosomes, so we're going to require those proteins. We're going to need the genes that code for proteins needed for DNA replication. If we are inhibiting their transcription, we're not going to have those genes available. We're therefore not going to be producing those proteins. Our non-phosphorylated form of RB is active. So it's active when it is not phosphorylated. And what it's doing is it's going to bind to a transcription factor and keep that inactivated. So it's suppressing this transcription factor, if you like, and stopping any progression from G1 into S phase. When we wind up with our cyclin CDK molecules around, this becomes phosphorylated. So our active RB protein becomes phosphorylated by those CDK proteins produced a little minute ago. So they've been activated by our cyclins. The cyclin CDK molecule is going to come down as activated and it will phosphorylate things. So it will phosphorylate our RB protein, and when that's phosphorylated, it's then inactive. That releases our transcription factor, allows the transcription factor to bind to the DNA, and allows the transcription of the genes required for the proteins needed for DNA replication, and allows the cells to progress from G1 to S phase. So at the G2 checkpoint then, we are looking for cell size again. We're looking for how much growth we've got. We're also looking for DNA replication and DNA damage. So we've just come out of S phase, then we're towards the end of our G2 second growth phase. So this is the chance that the cell has to check that DNA replication has occurred successfully in our S phase. The DNA damage, if there is any, can trigger the activation of several proteins, and their options are to stimulate DNA repair, stop the cell cycle, or to cause cell death. Now, one of the proteins which is activated as a result of DNA damage is one called P53. So if there's any DNA damage, it moves from its inactive form to its active form. The P53 protein is quite a important protein. It's been described previously as the guardian of the genome. It's got a role in really maintaining a functional genome, making sure all the DNA is correct. And a lot of human cancers actually are linked to mutations in this P53 gene. And that sort of tells you just how important that protein is in order to regulate the cell cycle. So if we do get any DNA damage, our P53 molecule is able to bring about a few different cellular responses. So it can activate particular DNA repair proteins, and they're able to repair the DNA and allow the cycle to restart. It can arrest the cell cycle at the G1 checkpoint, and that means that the cell cycle stops, allowing the DNA repair proteins time to recognise and fix the DNA. And if the DNA damage is too severe, it can initiate apoptosis or programmed cell death. Our final checkpoint is the metaphase checkpoint. And at this point, we are looking for chromosome alignment and spindle formation specifically. So the metaphase checkpoint isn't just in mitosis, it's really controlling the progression during mitosis from metaphase to anaphase. So we're checking to make sure that the chromosomes are all correctly aligned across the metaphase plate. And this is making sure that really they're aligned in the right direction and that the sister chromatids are paired together. We're also checking to make sure that we have correct spindle formation and that each spindle fibre we've got a spindle fibre attached to the kinetochore between each sister chromatid. And this checkpoint is really making sure that each daughter cell is going to receive the correct number of chromosomes. So during our metaphase checkpoint, we're checking to make sure that those chromosomes are aligned across the middle and that we've got a spindle fibre connecting to our kinetochores, allowing to make sure that we've got the correct number of chromosomes on each side. So it's quite important that the rate of cell cycle is tightly controlled. If we reduce the rate of the cell cycle, we may result in degenerative diseases. For example, alteration in control of the cell cycle can occasionally lead to the expression of proteins which are associated with Alzheimer's disease. Alternatively, an increase in the rate of the cell cycle may result in tumour formation, and this can also be caused by proto-oncogenes. Now, a proto-oncogene is a normal gene, which is usually involved in control of cell growth or cell division, and occasionally it can mutate to form a tumour-promoting oncogene. So those oncogenes are going to cause the cell to divide in an uncontrolled and unregulated manner. So it may continue to grow and divide despite having the appropriate growth signals given to it. And mutation of just one oncogene to an oncogene within a cell is enough to cause unregulated growth. So from that, you should be able to explain that the checkpoints that are present within a cell are mechanisms within the cell which are going to assess the condition of the cell and halt or promote progression to the next phase. That we have a number of different proteins involved, such as cyclins, cyclin-dependent kinases or CDKs. We've got the retinoblastoma protein, and we've got the p53 protein. That at the G1 checkpoint, we have the activation of CDK proteins with cyclin, and that they phosphorylate target proteins, and that phosphorylation will allow the cell cycle to move on to the next stage. Partly as a result of this, the phosphorylation of retinoblastoma protein or RB protein will deactivate it and prevent it from binding to a transcription factor. That transcription factor can then go ahead and activate gene expression to produce proteins involved in DNA replication. During mitosis, we have the metaphase checkpoint, which is essentially making sure that we have progression from metaphase to anaphase by ensuring we've got chromosomes aligned correctly and attached to the spindles. And finally, we may have problems caused by uncontrolled cell division. If we have too slow a cell cycle, we may result in degenerative disease, and too fast a cell cycle may result in tumour formation. A proto-oncogene can be mutated to form a tumour-promoting oncogene, which will cause the cell to divide in an uncontrolled and unregulated manner. As ever, feel free to ask any questions at all on this as we go over it in class.